Well, thank you very much indeed, Valerie, and to the rest of the committee, which I am now no longer part of, for the very warm, kind invitation to speak to you today about what I think is a really exciting emerging technology. And I say emerging because when I heard that 200,000 people have been implanted with DBS devices in Peter's talk, it made me realize that probably only a few hands are needed to count the number of people in the world that have been stimulated with sound through for neuromodulation. And so just to give you a little bit of background about why I'm interested in this, it all started with a pint in the Crown, which is my local village pub, with my good friend Robin Cleveland, who's a professor of bubbles at Oxford University, a professor of ultrasound engineering. And I was talking with him, our kids go to the same primary school, and I was talking with him about the fact that I am interested in memory and memory dysfunction in the hippocampus, and bemoaning the fact that non-invasive neuromodulatory techniques such as transcranial magnetic stimulation are not able to reach deep into the brain and in a very targeted way. And he told me that using ultrasound, people had started, at least in animal models, to be able to direct ultrasound waves across the skull through the bone, which is a barrier to ultrasound transmission, in a very focal way and affect neuromodulation. So we became engaged in this study. Our kids were at that stage at primary school together. They are now doing their A-levels together. And we've completed one experiment, which this is how things go when you're working on a shoestring. So I must warn you that this is very much an emerging technology again, and thus there will be limited clinical content to this talk, but I do hope that we can somehow pique your interest in this to something which I think probably will have quite a significant translational application around the corner, both for research and treatment in neuropsychiatric disorders. So this is one of those PubMed graphs, just to show really that in the last few years there's been a massive increase when you put focused ultrasound and brain in the number of papers that are published per year. If you put neuromodulation in here, because there are a number of different applications of ultrasound in the brain, then the number goes down quite considerably. But I will talk a little bit about some background on ultrasound briefly, talk a little bit about some of the other animal and human studies of neuromodulation using ultrasound. I'll describe our own recent experimental results, and then try and bring in a few clinical applications that are emerging of this technique. So ultrasound, the term ultrasound denotes sound that has a frequency above the human hearing range. Humans can hear really between 20 hertz and 20 kilohertz of sound. Dogs can hear up to about 40 or 50 kilohertz. Bats can hear about 200 kilohertz. And diagnostic imaging, which has obviously been around for a long time with ultrasound, uses really high frequencies in the several megahertz range, because at those frequencies you get very high spatial precision of the ultrasound beam. Neuromodulatory techniques use slightly lower frequencies, around 250 kilohertz to about 2 megahertz, because these frequencies are better able to pass through the brain, through the skull. So ultrasound was first sort of not invented, but looked at by Francis Galton, the 19th century polymath who invented the Galton whistle, and he used this to probe the hearing ranges of different animals. But nowadays we use the piezoelectric effect, which you may well have heard of. These are dipolar structures, crystals like quartz, which deform when an electric current is applied across them, and can be made to vibrate at a very high frequency by alternating current. And the effect also works the other way around, such as when that crystal is squeezed or stretched, it generates a voltage across the opposing surfaces. So this effect has been used for a long time in ultrasound imaging, in which the ultrasound transducer here emits a very brief pulse or ping of ultrasound energy, And then different tissues have different acoustic impedances according to their density. Okay, and at the boundary of two different tissue types, two different soft tissues, for example, you get a reflection of a certain percentage of the acoustic energy, and that reflection can be detected by the very same transducer using this piezoelectric effect. And the time delay obviously reflects the distance, and the image is computed. Now, bone has a very high acoustic impedance, so almost everything is reflected. And that's why ultrasound is very good for imaging soft tissues like babies. Now, note that a coupling medium, such as a gel or degassed water, is needed between the transducer and the skin to avoid reflection at that air-skin boundary. Okay, so that's why you use this gel to allow transmission of the ultrasound waves into the body. Okay, so using curved transducers like this here, ultrasound can be focused at very high energy levels into intracorporeal targets. This is called HIFU, high intensity focused ultrasound, and was first pioneered back in the 50s by the appropriately named Fry brothers at the University of Illinois. So this tissue heating effect can be used to ablate regions of the body and is particularly developed now for cancer treatments in prostate, in liver, for bone pain, in pancreatic tumors. So this is very much a thriving field at the moment. Besides the thermal effect of ultrasound, there's also a mechanical effect simply due to the vibrations in the tissue medium which are produced. And that mechanical effect can be used therapeutically to break up solid things like gallstones and kidney stones in lithotripsy. And because of the bone reflection problem that I mentioned earlier, ultrasound has not really been used for looking at or causing lesions in the brain because of the skull, until very recent developments in technology have helped. And that includes, for example, these arrays of about 1,024 different transducers arranged into a helmet, this exablate system here, whereby pretty weak ultrasound fields, once they cross into the brain, can be focused together at a single point in space, deep in the brain, for example, in the thalamus, and used to cause very small lesions for the treatment, for example, of essential tremor. The temperature of the brain region that's being targeted is monitored using MR thermometry, and it's basically taken up to about 60 degrees centigrade, and the lesions are anywhere between 1 and 2 millimeters, so very, very focal. A major trial back in 2016 in essential tremor patients, in just over 70 patients, showed that this was remarkably efficacious and also safe, and is now in routine use in several countries, including the US and Canada, for moderate and severe essential tremor. It's not yet NICE approved in this country. The advantages are that it's non-invasive, unlike DBS, for example. It involves no ionizing radiation, like gamma knife surgery would do. And you can monitor the pre-ablation efficacy as you ramp up the temperature. Before it causes a thermal lesion, you can detect whether or not there's a clinically significant benefit before you actually cause the lesion. And also, once you've forked out the 1.5 to 2 million pounds necessary for the helmet, the ongoing treatment costs are relatively low. This is now being used for trials in other movement disorders such as Parkinson's disease, and also for psychiatric disorders in OCD and major depressive disorder. There are trials ongoing with lesions in the anterior limb of the internal capsule. But going back to the Fry brothers, and because this session is about neuromodulation rather than lesioning the brain, I'd like to tell you about this effect, first of all, that was first observed in 1958. They took some cats and made a window in the skull, and they found that by stimulating with ultrasound, focusing ultrasound on the lateral geniculate nucleus of the thalamus, they could attenuate the visual evoked potential. So just a few seconds of stimulation. This is before stimulation, this is during stimulation, and this is about 30 minutes after stimulation where you get that effect is then resolved. So more recently, people have used pulses of ultrasound to modulate sensory and motor cortex in rodent models and demonstrated highly focal neuromodulatory ability of ultrasound. So for example, these movies here from Sung-Shik Yu in Harvard show, as the ultrasound is on, twitches of especially the right forepaw, and in this movie here, of the tail, with each ultrasound burst. So how does this happen? How can sound trigger or modulate the function of neurons? Well, hippocampal slice preparations show that you do get action potentials here with ultrasound stimulation, and these are sodium channel dependent. If you block the sodium channels with tetrodotoxin, those the action potentials disappear. But how do you get action potentials? And this is the subject of considerable investigation at the moment. In contrast to the high Fu, these low intensity techniques have very low thermal energy, so temperature rise is thought to be negligible, and indeed if you knock out thermosensitive ion channels in the preparations, then the effect continues. Whereas if you knock out mechanosensitive ion channels, then you still get action potentials generated by ultrasound. So the most simple hypothesis would be that acoustic pressures generated by the ultrasound actually cause shear waves here which travel through the phospholipid bilayer of the membrane and directly affect mechanosensitive ion channels in the membrane, okay? Causing conduction of current. An alternative is important for reasons I'll talk about later, that ultrasound can cause something called cavitation. So you can get little pockets of dissolved gas, or and particularly you can get this within the lipid bilayer of little tiny nanobubbles that expand and compress with the ultrasound pulses. Okay, and that can build up over time, and you can build up a capacitance current, current across the membrane, and that can eventually result in action potentials. Okay, so turning to human studies, there have just been a handful of these over the past 4 or 5 years, where people have tried to target focused ultrasound for neuromodulation, and this is using single ultrasound transducers, like in the rat you just saw, rather than the complex helmet, to the primary somatosensory cortex, primary motor cortex, visual cortex, and some deep structures like the thalamus. And I'll briefly describe 2 studies before going on to describe our own. So this is one from Win Legon at Virginia, who I'm not sure if you can quite see, but using a 0.5 megahertz ultrasound beam, modeled this and showed that at primary somatosensory cortex here, S1, he could get a sort of cigar-shaped beam of ultrasound with a 0.5 centimeter diameter by about 18 millimeters, 1.8 centimeters in length. And this beam, when targeted at S1, attenuated the somatosensory evoked potential when people had somatosensory stimulation to the hand. Okay, so the SEPs were attenuated, as you can see in both of these figures here, by the ultrasound compared with sham. And when they did that, that actually led to an improvement in patients, healthy subjects, ability to functionally discriminate between 2 points or 1 point in the hand, or the relative frequency of a stimulation. So their ability was improved by this cortical suppression by ultrasound. Another study from Sung-Shik Yu in Harvard was very striking. They managed to stimulate V1, primary visual cortex, whilst subjects were lying in the MR scanner. So very technically challenging, and at the individual subject level, they showed activation throughout visual pathways in every single subject. And in fact, there were more subjects than that. There were about 16 subjects. 5 of them did not experience phosphines, and these 11 did experience phosphines. And the 11 that experienced phosphines had changes in their visual activation, whereas those who did not have changes. Okay, so in order to sort of spread out these, to have more widespread human studies, the safety profile of this technique has to be clearly established. So we just need to understand a little bit about the way ultrasound energy is delivered to the brain. Gosh, it's counting down. My time is counting down. I thought it was counting up. Right, let's not talk about safety. Doesn't really matter anyway. But basically, the important thing to say is that the existing guidelines for ultrasound safety are out there for diagnostic imaging, okay? And the FDA levels are up here, and you measure, basically, you take into account the amount of pressure that is generated by the ultrasound. There's a thing called the mechanical index, and the amount of temperature rise which you get as well. And all the studies so far, including our own in light blue here, are well below those levels. However, the way in which ultrasound is delivered is very different from imaging. So as I said, in imaging, it's a single pulse, you wait for it to come back, and then you scan somewhere else, and it's a single pulse, and you wait for it to come back. Okay, and the frequencies are also much higher, whereas within stimulation, we're using, your 500 kilohertz ultrasound is given in pulses, each of which lasts about 0.5 of a millisecond, and then you get, those pulses are repeated in a burst. So you get little bursts within bursts of ultrasound that then go on for, well, in our case, 300 milliseconds, sometimes up to 40 seconds. Okay, but we've surveyed the literature of about 58 papers in animals and the humans, and there were really no safety concerns out there, and we managed to convince our IRB that that we were able to go ahead. So we decided what we wanted to look at was higher visual function. Okay, this is Robin, my engineering colleague. This is Joe Blackmore, who was his PhD student, and Verena Braun, who was my postdoc. This is our transducer that we used, which has a fixed focal length of 10 centimeters, and it's stimulating at 500 kilohertz. What we did was we were really interested in the area MT, which is responsive to visual motion. So we put our 16 subjects before the experiment into the fMRI scanner. We found their MT their individual MTs by showing them moving dots. We then segmented out their brain, and using the segmented brain with all the different types of tissue, skull and CSF and water, we were able to create computer models whereby we could determine exactly where we needed to place our transducer in order to hit that spot and to deliver safe amounts, what we hope are safe amounts of energy to the required site. Okay? And the way you do that is you basically, you imagine you've got your source here inside transmitting ultrasound out. You collect what is out there where you might place your transducer, okay, and you measure the phase distribution and the amplitude distribution there, and then you reverse model that to make sure it's hitting the right place. So this is our phase distribution as we imagine we're at MT, and you can see there's an area here where the phase is relatively homogeneous, and when we refocus we get this nice cigar shape, again with about 5 millimeters across. Okay, when we tried to do deeper targets, the field becomes more fragmented, so the hippocampus still needs a bit of work. So this is our experimental setup. Here's Robin stimulating a subject. We have the cone here. It's connected to the skull with a water-filled membrane. It's very important, as I said, to get good contact with the skull, so with the skin of the scalp, so that that ultrasound is transmitted clearly into the brain. When Robin turned up, he was the second person in our study to actually undergo the stimulation. I saw him in the morning. When he turned up, he had shaved his entire head with a razor so that we got good contact. Not everyone in the study did that. So we also have EEG monitoring, and both the head and the cone are located in space by an optical tracking camera, and we can then combine that with the MRI scan and know exactly where our ultrasound waves are going in the skull, or at least model that. Okay. So here we are again. There's our bursts of ultrasound. This is the task we use. So there's a period of fixation, you then get, it's basically you have moving dots that are moving at random, and then a certain percentage of those dots you move either to the left or to the right. Okay, and each individual person will have a threshold at which they can detect whether those dots are moving left or right, and their job is to guess whether they're moving left or right. And we find each individual's threshold where they're 70% accurate, and we work around that threshold. Okay, so they fixate, they see the dots either on the left or the right side of fixation, then they make a response whether it was moving left or right. Now, the first thing to say is that subjects were not able, the other thing to say is that we didn't, in order to get the spatial specificity, we also stimulated at, as well as stimulating at MT, we stimulated at another site, the fusiform face area, to see whether that also had an effect on movement perception, and we hoped that it wouldn't. So when stimulating at either of those sites, this D' of 0 is chance level. Subjects were not able to determine whether or not on any individual trial they were being stimulated. Okay, the really reassuring thing and the surprising thing was that we actually did detect a difference between stimulation and non-stimulation trials, and similar to the other studies that have been published in other domains, we noted facilitation of movement detection with stimulation. Okay, so this is stimulation on the filled squares. This is the left visual field, and that's visual field, and that's the right visual field. So this is the contralateral one. We always stimulated on the right side. This is the contralateral one, and the one where we would expect to see an effect, and that indeed we did. When we stimulated at FFA, so the other deeper site, we did not notice any difference between stimulation and non-stimulation trials. And indeed, a direct contrast of the MT versus the FFA stimulation confirmed that this spatially specific effect was just for the contralateral hemifield and not for the ipsilateral hemifield. We were so surprised by this result that we actually reran the entire experiment again on all 16 subjects, and found exactly the same result. So this is, we're very convinced that this happens, even though it's a relatively mild effect. Looking at the EEGs, across the whole 300 millisecond stimulation period, we found that there was a significant difference between stimulation and no stimulation trials, particularly in this cluster here in the left frontal lobe. The ERPs, this is 0, this is when time lock to the onset of stimulation, and the first 300 milliseconds of stimulation finished here, but also during the fixation period after that, there was a difference in the ERPs between stimulation and non-stimulation, and that was significant only for MT and not for FFA. We did look at safety. None of our subjects described any discomfort or any ongoing problems or any problems at all with the stimulation. And we did repeated imaging after stimulation, and that showed no abnormalities at all, no difference between the pre and post scans. Even with, this is SW, susceptibility weighted imaging, which we included because there were just a few hints in the literature that some animals had microbleeds, microhemorrhages after this procedure. We didn't see any of that, and I think actually the animal literature is probably artifactual. So, we're very excited about these results. It's amazing that sound waves can be targeted so precisely to change both behavior and physiology across this barrier of the skull. The spatial specificity of this technique is much better than other existing non-invasive brain stimulation techniques. It's very early days, but there's a lot of potential clinical translation and talk about work in Alzheimer's disease, Parkinson's disease, epilepsy, depression, and chronic pain. Partly because of other biological effects, which I'll just very briefly mention over time. But one of the really interesting things, I talked earlier on about cavitation. So you can actually inject microbubbles into the circulation and induce this cavitation effect within the brain by focusing your ultrasound. And that opens temporarily the blood-brain barrier and can allow drugs across the blood-brain barrier in a very spatially specific way. This has been used, for example, in patients with glioblastoma. You can't really see it back there, I suspect, but there's leakage here of gadolinium at the place where the brain has been sonicated, and that's the same here in this patient who's got Alzheimer's disease. So this is a very exciting way of potentially delivering very focal therapeutics across the blood-brain barrier. In the future, we need better delineation of safety parameters. We need to be able to localize in real time where our ultrasound stimulation is happening, which is difficult at the moment. It was all done with modeling, but there are emerging MR techniques where you can actually see those tiny nanometer vibrations of the tissue caused by ultrasound. You can visualize that. That's not yet possible in humans, but those techniques are undoubtedly going to emerge soon. There will be the possibility for multi-transducer arrays to target even more precisely and potentially target multiple locations at the same time. And wearable or implantable devices may well become possible as well. So there's evidence, again, from animal literature that rats with epilepsy, if you focus ultrasound on them, then the seizures are prevented. Okay, so it can modulate the neuronal activity in that way. So these are all soon to come applications in the clinical world. And with that, thank you very much for your attention.
Thank you, Chris, for this excellent presentation. Do we have any questions from the audience?
Thanks so much, Chris. That was so exciting. I'm just curious if there's any way of modulating the amount of ultrasound in the way that we give people different TMS amounts, depending on like MEPs. So duration
you mean
or frequency.
For individual thresholds of sensitivity?
Yes.
I mean, there should be. We don't have that yet. We don't. The effects are so small, you can't generate. No one's yet been able to generate a motor response in a human. So we can't get that sort of motor threshold that you'd use, for example, with TMS.
What about
but there may be a way of.
phosphines?
I don't believe. I think the phosphine literature is. Not many other people have been able to get phosphines apart from that one group. We couldn't when we targeted V1. And it turns out the way they asked about phosphines was quite leading. So I'm not sure. What could happen is you can combine TMS and ultrasound. So you can adjust the TMS, you can change the TMS thresholds using ultrasound with the motor thresholds. So that may be a way actually of finding those sort of thresholds that you're talking about.
Yeah, good idea.
Yeah.
So Chris, well, back here, way back. So insider knowledge, one thing they might find interesting is the auditory effect and what you had to do to get the experiment to work, just in terms of some of the side effects.
Yeah, okay, great. So I can tell you about that. I didn't include it just for time's sake, but so I mentioned that the ultrasound is delivered in pulses. So although the ultrasound itself is at 500 kilohertz, which is way above human hearing, it's delivered at effectively 1 kilohertz because you have these 0.5 millisecond pulses. Now, when we first ran this experiment, we found that people were able to detect the trials on which they were being stimulated and those that they were not being stimulated. And the reason, they all said, was because they could hear this sort of background noise during the stimulation trials. They initially just discounted that. They thought it was out there in the room. It didn't sound like it was in their head. It was out there somewhere. And it was pretty subtle. But we did various experiments using a skull in a water tank and our transducer, and we found using a recording device that we could actually pick up sound waves traveling through the bone at precisely that frequency, at the 1 kilohertz frequency. So the idea is that envelope in which the ultrasound is delivered is causing shear waves through the bone, which are then picked up presumably by the cochlea directly. Now, this has never been discussed in previous human studies. It must be there, and it may well be biasing some of the effects that are out there in the literature. But we managed then to counteract that simply by giving subjects little earphones and playing a tone on every trial, the stimulation and the non-stimulation trials, at that 1 kilohertz frequency. And as I showed you, those D prime values were 0. So with that, we're not able to detect it. So that's going to be very important in future work.